Okay, are there any questions? If not, let's go ahead and get started. Can somebody confirm hearing me?
We can hear you.
Okay, great. You should be able to see the screen now. I don't think I need this or that. My computer is going to be slow because I'm processing the converting the meeting recording for the lecture. So let's see. Need to go to modules. I think it's week 5. Actually, I'll start there. All right, so today we're not going to do the lab. I'm going to discuss the start of the unknown project, and hopefully, keep my fingers crossed, I will be able to mail out the start of the unknown project tonight, so that we'll say that the unknown project is starting on May 3rd. Any questions about that? Obviously, I haven't mailed it out yet, but I'm hoping to mail it out tonight, and then we'll start the unknown project, tonight. There we go. Any questions about what we're doing? Somebody had a question about the lab. Do you want to ask it now? I didn't do that. Let me get rid of there. I think I already saved it, but all right, if there's no questions about what we're doing, I'm going to close this down because I've got a lot going on in my computer. And let's talk about the unknown project. I should probably download this. And then it will display better. Let's go back here. So you can find the instructions under week 5 of the lab. And the first is actually lab 7, which we'll cover on Thursday. In the unknown report, the first link is a sample journal. It would tell you what your unknown report should look like. It actually gives you an example. This is the instructions, building a dichotomous clan, and then the unknown list. We'll want that later. And I'll open those in a little bit. We'll talk about those later, but I've got too many things going on at my computer. Once this stops, I'll have more resources on my computer. My computer is a little slow at the moment, so I don't want to open too many things up. Although I can shut this down now. Let's go ahead and shut those down. Turn that down too. All right, so the unknown project is worth 75 points. It'll be starting from today and will be done near the end of the term. I've got it here. It is due, I think, June 11th. Scroll down to the end here. Yeah, unknown reports due June 11th. Any questions about any of that? You can read about the introduction and the rationale. What this will be is a an unknown project, we'll all be giving you a bacteria over the computer, meaning we're going to pretend that I'm giving you a bacteria. I'll simply give you a number. And then you're going to be running different tests on the bacteria to identify it. And there's a large number of tests you can use to identify the bacteria in the lab, week 5 lab unknown test. All of these tests and a few others you can run. Any question about that?
How many tests are we supposed to be running in week 5 and the subsequent week?
I'm sorry, I can't hear you.
How many tests are we supposed to be running in week 5 and subsequent week?
Generally speaking, you'll only be running one test at a time. That's using dichotomous logic. Once you get done with that test, you can request another test. And let me go ahead and open something. Oh, let's see if I can. I'm just going to open it here. It'll display better if I open it here. So to identify the unknown species, on the left is all the possible species. On the top is all the tests, or most of the possible tests that you can run. Why did that roll down? So I don't know how many species are up here. These are the gram negatives. Maybe there's about 40 species. These are the gram positives. And these are the various tests you can run, going across the top. And it ends with motility. And then these are the different results for the different tests. For example, Citrobacter furendi is gram negative, its shape is bacillus, its growth arrangement is its singles and pairs. You can find it that it does not hydrolysize gelatin, and that's hard to read there the way that is. Let me shrink that down. Ah, there, that's too small, but you can see it gelatin there. I'm going to bring it like this, then I can more easily see it. 6.5% sodium chloride is negative, 7% sodium chloride is negative, nitrate reduction is positive, catalase is positive, etc. Any question about any of that? So this chart, which is shown right here, is the unknown list. It has the possible bacteria species and then the results to the different tests. And you can find that list here. Any question about any of that? So when you get one test done, you can take a look at this chart. Let's say you figured out it's gram negative and you figured out that it hydrolyzes gelatin. Then it narrows it down to, I can't use the color in this for some reason. Come on. I told you my computer is slow at the moment. There we go. So it narrows it down to 4 possible species. So you can find a test to separate these 4 possible species. And here's one. This will split it. What's a better one? This test could also split it, but it's separated into 3 and 1. I'm looking for one that separates it into 2 and 2. Well, I'm not finding one off the top of my head at the moment, but I'm sure there is one. Just look through the chart and you can find one. one that separates these right here. Well, I'm not going to spend any more time on that, but find another test to divide these for. And I've mentioned that this would be one test that could do it. And there are other possibilities too. And then ask for that test and you'll get your results. And if it's negative, then you got your species identified. If it's positive there, you've narrowed it down to 3 species now. Any question about any of this?
Yes, I have a question. So when we send you an email, do you want us to lay it out in a certain format?
I'm not hearing you too well. Can you guys speak up? I'll turn up the volume further. Can you repeat that?
Yeah, sorry. Do you, when we send you the email, do you want it in a certain format?
You're not going to be sending me an email. I'll explain how you're going to be requesting this. Now, why did that pop up? I certainly didn't want that up. And
Like
it
when
doesn't-
asking about what test we want to run.
Yeah, you don't do it in an email. I'll explain how you do that in a little bit. Okay, you will do it in the unknown notes. Let me show you. And this page here, unknown notes, you'll be doing it in your unknown notes page. So you'll be requesting everything in your unknown notes page. The reason is that way everything will be together. And then I will give you your test results in the unknown notes. Let me open this page up just briefly. I've got too much open here. Let me close that down. I want to keep that open. One of these I want to keep open. I don't remember which one. In your unknown notes page, you can, you've got a link here for, what is it called? Uploading a page. Put everything on your page that you're uploading and you'll have a link there. I don't have that because I'm an instructor. I've got the speed grader, which will allow me to access the page, and there's not going to be anything up here because nobody submitted anything. But you will have your information here in a page that you submit. Put everything in the page. Do not put anything in the comments because I'll be adding comments and I will be giving you the results in comments. And as you see, there's not a lot of room over in the comments. And so if students have a lot of comments, you're going to have to scroll down pages and pages of information to get your information. And I don't want to scroll down pages and pages of information to do that. So put all of your comments on the end of your page that you submit. And you can submit an unlimited number of pages. You'll have a link here for doing that. I don't have it because I'm an instructor, but like I said, I'll give you the results for a test that you have in the comment, and I might give you another comment about what you're doing or how you could improve or answer a question you have in your stuff. So everything you do, submit as a page in the unknown notes. Okay? And we'll come back to this later. So I'll leave it up because I really haven't talked about the unknown notes yet. I've only talked about the test and then all of the species that you can possibly be. Like I said, I'll give you a number. The number stands for one of these species, and then it's your job to identify the species. For example, if yours was gram-negative, was a bacillus, was gelatin positive, it's down to 4 species. And then if you did the urease test and it's negative, that means you have Salmonella triferium. So that would be your unknown. All right, let's go ahead and talk about the project. So everything we do will be done online, but we're going to do it in the sense that is if we are actually doing it in the lab. So phase one, you will be given the bacteria, and we're going to pretend that you're going to streak it out to isolate it. And you have to streak it out twice on a plate to make sure it is a pure culture. In the lab, we normally give you a tube that has 2 bacteria in it, and you have to streak it out to get a pure culture. So you streak it out and get a single isolated colony, take cells from that single isolated colony and streak it out again to get a single isolated colony. And after it's ran through 2 colonies, you then pick the cells from a single isolated colony and grow it up. And that will be your stock culture. Any questions about any of that?
So I'm confused, and this may be a dumb question, but are we doing this weekly? Are we till the end of June when it's all
No,
due?
you're not doing it weekly. You're doing it from when you get the test results. As soon as you ask for another test, within a day or so, I will start the test, and then the test will incubate. And let's go here. And you can look up what the procedure is for the test and how long the test will incubate. Most of the test will require 2 days time of incubation. Some of the tests, only a few, you can get the test on that same day. So if you have catalase cells and you ask for the catalase test, you can get your results on the same day you ask for it. But there's very few tests that you can get the results on the same day. There's the oxidase test that if you have fresh cells, you can get the test on the same day. Catalase is not particularly picky about whether the cells are new or old. Oxidase is, and you must have fresh cells. So if your cells are older than 2 days old, I will allow it to grow for a day before giving you the test results.
So are you gonna give us all of that information in our
No, that
email?
information is here. You just click on the catalase and it tells you how to do the catalase test. So you can look it up, okay? I'm telling you, most of the tests take 2 days to incubate. And after incubation, I'll give you the results. Some of the tests take longer. The MR test takes 5 days to incubate. The gelatin hydrolysis test takes a week to incubate. Okay?
Okay,
Most
so
of the tests take 2 days.
maybe I missed this part, but what is all included in the email you sent to us?
You're going to be writing stuff in the unknown notes page. You're not sending an email. Okay?
No, but
And I haven't talked about how you're submitting stuff.
But you
So
said-
let me talk
Information?
about that in a little bit. Okay?
I have a question too.
Okay.
Do we need to request, do we need to let you know what test we are requesting?
Yes.
And
I would not be able to read your mind, so you got to request a test.
through unknown notes?
Through unknown notes page, yes. All right. Let's.
I
go
have
back
one
to
more
the
question,
project.
Dr. Volt.
What?
One more question. So I may be misunderstanding this, but that entire list of tests that you just displayed in your screen, are we completing each and every one of those tests?
No.
Okay, that's what I'm confused about.
Yes. You're only going to be doing the tests that you need to do. Like I stated, if you know yours is gram negative, you know it's a bacillus, and you know it's a gelatin hydrolysis positive, and then you request the urease test and it comes back negative, you have identified your unknown species. Because there's only one species that can have those results. Okay, all right.
One more question. Would you be able to tell us if we are on the right track, determining the unknown, or if we have identified it correctly?
No, I'm not going to tell you whether you're on the right track, meaning you're successfully identifying the species until it is graded. I will tell you if you're making no sense in your, I don't know, your procedure you're doing, I will tell you, like, I don't understand what you're doing because you're not following, I don't know, dichotomous logic, you're not following the procedure, and you should reread the instructions. You guys have supposedly read the instructions. I'm trying to talk about it now, okay? So first, I will give you the species, and this is going to be pretend, where you're going to pretend to isolate it. Normally, you have to go through it in the lab, and you actually do have to isolate it because you will be given a tube that had 2 species in it. However, because we're doing this online, I'm going to give you one number, and you're going to pretend to isolate it, meaning make sure it's pure. You're not actually going to be doing it because it's done online. Okay. But when you write it up, you should go through and say on May 3rd, assuming I send out the email tonight, that you got your unknown number and that you streaked it out on a tryptic soy agar plate or a NA plate to isolate colonies for the purpose of making sure it's pure. And then you got one isolated colony and you streaked it out a second time to make sure it's pure. And even then it's not 100% guarantee it's pure. It's only 95% guarantee or greater that it's pure. And then you use that to set up your stock culture. And that's the species identification. And the good news is when I give it to you online, I'm just going to give you one number. Okay, so there's not really any reason to streak it out, but you should state that on May 3rd, you got the unknown number, and then you streaked it out twice to get isolated colonies to grow up your stock culture. Okay, so you should say that in your unknown report. All right, and then you're going to go ahead and run the test. And I showed you the test that you can run. I also mentioned that you should be using dichotomous logic to run your test. Dichotomous logic means you will run one test. Let me see if I got a picture of this. This is close enough. Well, no, it isn't. Here's dichotomous logic, sort of right here. You have different possible bacteria, and these are the different cellular morphologies. You can have rods and cocci. And so you can do the different shape, and that tells you whether it's a rod or a cocci. Okay. You can then do the PR glucose test, and let's say it's a cocci, and the results come back PR glucose positive. So we're only looking at this result and this result and this result. So that means this one is not under consideration. Let me remove the highlight there. the next test, oops, that didn't work. The next test you run could be the PR lactose test. And if it's positive, this one is removed, and this one is the species. Okay, so that is using dichotomous logic, where you're running one test at a time, and you are you have two possible choices, positive or negative, or in this case, you had cockeye or rods. Any question about that? That is using dichotomous logic. If you don't use dichotomous logic, then your project will be penalized. So you want to use dichotomous logic. Where was I? One thing you could do, gosh, why did that move there? Probably because my cursor was there. You could run a whole bunch of tests at one time. That's called the shotgun approach. And you will be penalized heavily if you use the shotgun approach. For information about determining your species, you can find information in Berge's manual and the lab manuals. You can get the Berge's manual and 1 or 2 lab manuals in the library. And I don't know how they do that, but there is some way for accessing the course reserve online. And I don't know how they do it, but there is some way to access it. And then you can actually physically go into the library and get it that way too. You can get Berge's manual. I think you can get it online, or you could get some information from online. But if you get your information from online, make sure it is accurate. And then of course, I'm giving you the test results so you can look at that table. And then I give you all of the tests so you can look and read about the tests that you want to run. Okay. All right. And then the first thing I need to do is I'm going to tell you that your first test, you're not going to be selecting. I am going to give you the first test to run. And the reason is because I looked at this project, and if we were to do it in the normal fashion, where you guys request a test, and then I give you the results, on average, you're going to be running about 5 or 6 different tests. And even if you're only running 4 tests, okay, which let me see how we did that. We did the gram stain, we did the shape, we did the gelatin, and the urease. That was 4. So actually that's going to be a minority of the students will only be running 4 tests. But even 4 tests, and there's 20 of you, that's 80 replies that I'll be giving to. And if it's 5 tests, which will be majority of the class, that means it's 100. And I just said 100 responses for one project is an awful lot for one class. And so what I'm going to do is I'm going to select the first couple of tests you're going to be running. You're first going to be running the gelatin test, and I will start that tonight. And this one will take 1 week long, which is why I want to get you started on the gelatin test early, because this one takes 1 week to run. And then you're going to be doing, let me see, you're going to be doing the gram stain on Sunday, but you're not actually going to look at the results until Monday. So you'll be setting up the slide on Sunday, but you will be looking at the results on Monday. Okay, and then on Monday, you're going to ask for the PR lactose test, which takes 18 to 24 hours to run. And so on Tuesday, I will send the class one email containing the results of the gelatin test, the gram stain, and the PR lactose test. And in one email, I will be sending out one to the entire class with the results and your number. Instead of sending out 60 different contacts in the what is that called? Something notes, the unknown notes page. And so that's going to reduce my contacts with you by 60. And that means on average, I will have 40 contacts for this one project, for this one class. And that's doable. 100 contacts, that's a bit too much for one class. Okay, but you will be using dichotomous logic because you're going to start the gram test, not the gram test, the gelatin hydrolysis test tonight. You'll be starting the gram test on Sunday, and you'll be starting the PR lactose test on Monday. And you'll get all of the results on Tuesday night. Okay, any question about any of that?
Are there
And
any reminders of
I'm
where
sorry?
or what? Are there any reminders of what times we need to do these?
I'm sending out the information. So there's no reminder you need to know about these 3 tests. And I've already chosen that the first 3 tests you're going to study are the gelatin test, the gram stain, and the PR lactose. And the gram test is just a very good one because it narrows down the bacteria into 2 large groups. The gelatin hydrolysis is a good test to start early because it takes a week for that one to run. And then the PR lactose is just a good test. It splits. Why is this moving? Something's funny with my computer. Oh gosh, why did that move all the way over there? The PR lactose test, it doesn't quite split the gram negatives 50%, 50%, but it does a pretty good job, and it's very easy because all of the AGs are on top, all of the negatives are below, so it's easy to distinguish. And then it splits the gram positives not quite as well, but it splits them pretty good too. So I just decided that was an easy test to run, and it's a good test to run too. And then after that, you will look at your results and determine which test to run. And you will put that in the unknown notes page. Okay. What you should do when you get your unknown notes page is you should record your unknown number. And then when I send you the results, you should put the results for the gram stain, the gelatin hydrolysis test, and the PR lactose test. And after your results, you should give your conclusion. So if the cells are purple and round, the conclusion is they're gram positive cocci. If the gelatin is liquid, the conclusion is gelatin hydrolysis is positive. Okay, so that's the difference between a result, that's your observation, and the conclusion. The conclusion is your interpretation of the results. Any question about any of that?
After we do the PR lactose test, then are you assigning us another number?
No, you're then going to look at your number, look at your test results, look at this table, and then you're going to request your next test in your unknown notes page. After my first email, which will contain the gram stain, well, actually, after the second email, because the first email will give you a number. The second email will give you the gram stain results, the gelatin hydrolysis, and then the PR lactose test results. Everything should be done in your unknown notes page. Okay, so everything you do on this project will be done in your unknown notes page. And if you request a test in the email, I'll tell you I'm not going to honor it. So put it in your unknown notes page. I want everything together in your unknown notes page. And that way, if there's a question, both you and I can look it up. Because everything will be there. I don't want to go hunting through my emails to try and find out what your test result was and when I sent the results and what the results were I sent. Because occasionally students misinterpret the result. And so I'd like to go back and make sure that I actually sent you the correct results. And so far, I've always sent the correct results. Okay. So I'd like to have all the information together.
If we have a question regarding the process of this assignment, are you wanting us to ask that question in the unknown notes or in email?
You should probably ask that in your unknown notes page. You can also ask it in the lecture in the lab. Like if it's something general and I can answer it just off the top of my head. If it's something else, what would be a good example? Like a question like, what's a V mean? I'll tell you, well, it's actually on here. Just scroll down. The V means the results are variable. Okay. ND, not determined. The parentheses and then a plus or a minus means that's the majority of the cases are minus. We're going to treat that as a minus, but realize that there are some, what do you call that? Some variants of Citrobacter frundi that are actually indole positive, but the majority of the variants are indole negative. And for this project, we're going to treat it as indole negative. Okay. A variable means that the result could be positive or negative. And I will only give you a positive or a negative. But if you look on the chart, it's variable. That means it fits both a positive or a negative. Okay. What else do I need to talk about? Not determined, treat it like a variable. It does on the bottom of this chart tell you a little bit about the gram stain, the cellular growth arrangement, the gelatin hydrolysis. This one here is in the procedure for gelatin hydrolysis. This has to be done at room temperature. You can't incubate it at 35 or 37 degrees Celsius, because if you did, you would liquefy the gelatin. And since we're looking whether the gelatin is liquefied or not, you can't incubate it in the incubator. Anyways, it gives you some information about some of these tests. That's not terribly useful there. But these are very important, this information here. All right, let's go back to the instructions. You should write everything when you get results down in your unknown notes page. So every day, if you're requesting a test, you should write that down in your unknown notes page. And you should also write down always your results, that's the observation, when you look at the tube, and your interpretations. Okay, so that would be your results and your conclusions, which are also called your observation, your results, and your interpretation is your conclusion. So if the tube is yellow, that's an observation. The conclusion is, I don't know, the tube might be positive. The projects were 75 points. It should be typed. You should use 12-point font. I have trouble reading, so if your font's less than that. I'm not terribly particular whether it's single space or double space, but if you do use double space, it takes up more room, so that's better for you. I don't think I have a limit on the number of pages, do I? Let me look a little closer there. Make sure I don't state that someplace else. It shouldn't be unreasonable length, meaning don't make it a 12-point page, but I thought there was a minimum length. I'm not seeing this here. I'm not seeing that now, so we won't look at that, and I don't see it. And if I see it, I'll find it and I'll tell you about it. You should have daily description in your unknown notes test, which will include the test you ran, the observations you had on that test, and your conclusions, your interpretation. Sometimes students just say the test was positive. And then I go back and I said, well, something isn't quite right. You got a question about it. And if all you write down, your test was positive, I will say, well, I'm sorry, I can't help you any further because you didn't write down your results. So always wrote down both your results, that's your observation, and your interpretation, that's your conclusion. And that should be written in the lab report, as well as on your unknown notes page. You should not omit data to make your project look better. Drawings, photos, and tables are very helpful. They are not required, with the exception that you must include near the end of the report a table identifying your unknown species. In this table, or it could be an, what do you call it, a flow chart, a table or a flow chart identifying your unknown species. In the table, you should have all the possible species, which this doesn't have, but we can assume that they have all possible species. And then they isolate it as gram-positive coccus. And so they have the possible species here. And then it's catalase positive. So they have the possible species here. And then it's PR mannitol negative. So that narrows it down to 3. PR glucose acid. So it narrows it down to 2. And then citrate positive. And so they've identified this species as Micrococcus varians. Okay. And then this person ended the test here. I will tell you that you may do one more test to confirm that your unknown is Micrococcus varians. This is optional. You do not have to do a confirmation test, but you may do one more test after you've identified your unknown to confirm that your unknown is Micrococcus varians. Okay? And assuming that it is Micrococcus varians, you would then end this project. All right? If it's not Micrococcus varians, you then want to look up your results to try and find out where you went wrong. All right, so that table is required, and it's going to be telling me, a table or a flow chart, telling me how you derived your unknown as being whatever species you're saying it is. So you should have up here the test you use that identified the species. And let's say you had another test here that did not help you identify the unknown, in which case you don't have to put that test up here. Like maybe gelatin hydrolysis gives you the same 4 species as catalase positive. Then you don't need to include gelatin hydrolysis because you're not using it to identify your unknown. Okay. If you do include it, it's not wrong. Just stuck it right here or right there. It doesn't give you any extra information. You do not need to give the step-by-step details of each test you run. For example, I know the steps of the gram stain. All you have to do is say on such and such a date, which let's say Sunday's about 5 days from now. So around 5/8, I did the gram stain. And on Tuesday, I got my results back. And it was, I don't know, gram positive cocci. So the results were, it was purple, spherical cells, the conclusion, gram positive cocci. All right, that's all you need to do. You don't need to tell me the step-by-step details of how you ran the gram stain. Your thoughts and words should be in your own words. If they're ever not, you have to put them in quotes and say where you got them from. Otherwise, it's plagiarism. Plagiarism, if you plagiarize on this project, you will be given a 0 and told not to plagiarize, okay? Most people won't plagiarize because you're working on this project independently, but if somebody were to give you, I don't know, some conclusion and you say, oh, that's very good, I want to use it, just put it in quotes if you use their exact words and say where you got it from, okay? Otherwise, you should paraphrase, change the wording to your wording. You're going to be having a paragraph, not longer than a half a page, talking about your unknown organism. And so you should have a literature cited page for where you got that information. So that's right down here. I don't even have that there, but, and then you'll have at least one reference for where you got the information for the organism that you're describing. And it only needs to be a paragraph, okay? Talking about the organism, like what diseases it caused, where you can find the organism, stuff like that. There it is, microorganism paragraph. Places it found, treatments you can use, interesting information about the organism. This microorganism paragraph should be at the very end of your paper. The only thing that'll be after it will be the references. In the literature cited page, you may use any common format, APA, or is it MLA format? Just be consistent. They should follow the correct format for a reference, meaning you should give the author, the year, the article name, and then the article source. If it's from a journal, you say the name of the journal, and then the volume number and the page number. If it's from an online web page, the source will be the address of the web page. If it's a book, it's a little different. I'm not going to talk about that. You can look up how to give a reference. All right, let's go into what else is required. I think that's it for what's required. Let me talk about the grading. Obviously, don't plagiarize. You can find this information in the grading sheet, but let me run through it here. You'll be graded on the completeness of the test. Each entry should include as much detail as possible, meaning you should give the date you ran the test. You should give the test name. You should give your results. You should give your conclusions. Okay. And you, in your unknown report, you should state what the possible species are under consideration. You might talk about a little bit about why you decided to run that test, what the possible conclusions are. You should be organized, you should be clear, it should be neat. We already talked about that. You'll be graded on your written communication. If the report is free of grammatical and spelling errors, you'll get full points for that. It should be easy to read, easy to understand. You should successfully identify your unknown. This is only worth a few points. I'll talk about that later. It's either 5 or 7 points. I'll talk about that later. It's more important the logic you use in identifying your unknown than whether or not you actually come up with your correct species. Proper laboratory techniques should be followed where points will be deducted. In this case, you just need to use dichotomous logic because you're not actually going to be working in the lab. All right, let me talk about the grading. You do not need to answer these questions. This is only questions to help you understand dichotomous logic. Nothing needs to be turned in about that. All right, any questions about the project? If not, let me talk about the grading of the project. You can find out the grading by looking at the grading rubric. It details exactly how the project will be graded. Assuming it's loading. Is this still running? No, I'm not sure why my computer is still so slow. All right, the utilization of resources. Did you use excess media or did you perform irrelevant tests? If you did, you will have a penalty. If not, you'll get 10 points for that. Was dichotomous logic used in selecting your tests? If dichotomous logic is used, you'll get 10 points for that. If you have one excess test, you'll be only getting 8 points. If you use a shotgun approach, you'll only get 2 out of the 10 points. Are all relevant tests used? If no tests were omitted, you got 10. If one test was omitted, you only get 8 points. And you might not know that, but I can look at the table to determine, was there a test that you could have ran that would have helped you identify your unknown? And most people get 10 points out of this one, okay? Occasionally students wait to the end before they request tests, And so they run out of time and they don't do all the relevant tests, in which case they don't get full credit. Very rarely does a student not run a test that they should have ran, in which case they don't get full credit. Were the correct species included at each step? If all the species are correct, you get 10 points, okay? So you do need to talk about when you're running the test on your unknown species, unknown notes page, you have to tell me what species are under consideration. And if you have all the species correct all the time, you'll get 10 points. If there's one species at one point which is incorrect, you only get 8 points. If 2 to 3 species are incorrect at various times, you only get 6 points. If there's over 5 incorrect species, which I never had a student do that, you get only 2 points. The completeness of the report, if all information is presented, you'll get 10 points. One instance of missing information, minus 2 points. Effective and clear communication. If the English, grammar, the spelling, and the punctuation are all good, you'll get 15 points. If there's a few errors, but I can understand your logic, you'll get 13 points. If there's some errors and your logic is not understood, you only get 7 points. So I guess this would be effective and clear communication, and I guess your logic. And then the species identification. If you get the correct species, it's worth 10 points. If it's within the same group, like you call it, oh gosh, I'm drawing a blank here. Micrococcus luteus, and it's actually a Micrococcus varians, that's within the same group. So that would be, oh, where was I? Sorry, I've lost my place now. That would be worth 7 points or minus 3. If it's outside the group, but you get the right gram stain, you'll get 5 points. And if you get the gram stain wrong, you're going to get 0 out of 10 points. Any questions about the grading? The total will be 75 points. All right, any questions about the project?
So just to clarify, are we getting our species when you send out the first email next week with the test results?
You'll get the first species tonight. So I'm going
Okay.
to try and send out number tonight.
Thank you.
Okay.
If we,
And
if
then in
we,
a week from tonight, assuming I get everything done, I'll send out your first results.
so tonight we're just getting the name of the species, correct? And
You're
that's
getting just
what we're
the
noting.
number for the species. Correct. Any other questions? All right, any other questions about anything else?
I do have a question. So I see in the modules that there's the unknown case studies available. You also mentioned that there was information on the particular tests, but I'm not seeing that unless I misunderstood.
All right, let's get out of here. Everything will be written in your unknown notes page. And I'm not going to talk further about that. And here we go. So what was your question again?
So in the modules, I'm seeing the case studies listed. There's like case study one,
No,
two.
the case studies will be for the lecture. You're looking, you need to look under Lab 5. I'm showing it now, week Lab 5. This has the sample journal that gives you an example. I should talk about that before I end this. There's your unknown instructions. That's the instructions for doing the project, which hopefully you've already read. This is for building a dichotomous game plan. And like I said, essentially, you're going to be asking for one test at a time. And if it only has 2 results possible, then they are following dichotomous logic. If you ask for one test at a time and there's more than 2 possibilities, that's still following dichotomous logic. But an example is actually the PR lactose test. There's 3 possibilities. You can be PR lactose negative, you can be PR lactose acid, and you can be PR lactose acid and gas. But that's still following dichotomous logic because you're only doing one test at a time. Okay. That's for the unknown list. The unknown notes page is where you're going to be putting everything. This is just telling you how the project will be graded. And for your different tests, you come under week 5 lab. Oh, you can't see this.
So yeah, that was my question. I don't see
That's what
that
your
part.
problem is. That wasn't.
Yeah.
You're supposed to be seeing that. I think I just made it visible to you now. Sorry, I was supposed to turn that on. I thought I had turned that on last week, and maybe that change didn't stick for some reason. Let me look and see if everything else is turned on. But thank you for asking that. That's why you couldn't see it. So go to the 5-week lab unknown test, and it shows you all the different tests that you can run.
I just checked and it's in there now. Thank you.
Some of the tests, like the McConkey's auger, is a test you could run, but it's not directly on the chart of the unknowns. And that's because McConkey's auger can give you, whether it's gram negative or gram positive. If it grows on McConkey's auger, it's gram negative. And if it's yellow, that means it ferments the sugar lactose. So that means it would be one of these 4. But you could also run the PR lactose test, which I guess I'm giving you. And that's probably why not too many people run the McConkey's auger. My point is it does have some tests that are not on this table. And you can run those tests if you want. That's the only one I remember on the top of my head that's not on the table. Let me briefly mention this one here, talking about the gram stain, the cell shape, and the cellular growth arrangement. That's what arrangement stands for. It does have it on the chart. Where do I have that? Down here. It tells you the arrangement in the gram stain from an auger culture. Chains break apart and cells clump looking like clusters. So it's safer to not use the growth arrangement as a guide only and not eliminate any bacteria. If using it, you must grow your cells in a broth culture to prevent the chains breaking and to prevent the cells from clumping. Okay, and that's stated here. That's not it. In the gram stain. It is usually difficult to determine whether cocci are in clusters or chains when viewed on the gram stain. This is because chains are broken apart and because cells tend to clump together when taken from a solid medium. So the take-home message is don't get the cellular growth arrangement from the gram stain. You need to grow it in a liquid culture before you can determine the cellular growth arrangement. All right, I think that's the only thing I need to talk about. Am I gonna go backwards? Here we go. This is actually two tests, but they're similar, so they're put together. Whether the cell can grow at 6.5% or 7.57% sodium salt media and sodium chloride. All right, any questions? If there's no further questions, I think I'm done with the lab.
I have one question, but it's not about the unknown project.
Okay,
For enrichment cultures, this is about lab 5.
huh?
For enrichment cultures, would you consider that to be a selective media or not?
It is not a selective media.
Okay, thanks, because the book says like a selective media, so I just wanted to get a clarification. Thank you.
Your book said enrichment media is a selective media? Oh, wait a
It
minute, wait a
says
minute, wait a
in
minute,
a
wait a minute, wait a minute, wait a minute. Let me think about this.
It just
An
said,
enrichment media isn't in lab 5. I'm pretty sure of that. Let me go in there. I'm thinking of enriched media. There's a difference between an enriched media with an ED and an enrichment media.
oh,
Okay, there is a difference there. An
Okay.
enrichment media is not discussed in lab 5.
got it.
An enrichment media is a selective media. You put something in an enrichment media when you want to increase the probability of seeing that it'll grow in that media, meaning it'll be selected for. So an enrichment media is selective, but an enriched media like blood agar is not selective.
Got it. Okay,
Okay.
thank you.
Can you tell me what chapters the actual quiz or that the quiz is on for tomorrow?
I discussed that in the start of the lecture.
I missed the start because of work, sorry.
Okay, just a minute. Let me go there. You'd think I'd have it memorized, but no, I don't. I think it's the end of chapter 3 and chapter 4 in the lecture. Yep, the end of chapter 3 and chapter 4, and then labs 5 and 6. And that quiz can be done tonight, starting at 8 o'clock. Somebody is going to take this quiz tonight because they're going to work tomorrow or something like that. And they were asking me to get the first pass of lab 6 graded.
Yes, that's me.
Is that you, Nav?
Yeah.
Okay, that's what I wanted to ask. All right.
Yeah.
I'll try and get that done.
I was waiting for you to finish this so I can ask you to grade
Okay.
that.
All right.
Thank you.
All right. I think we're done here. Is that right? Anyone have any other questions? If not, I'm going to stop the share and end the meeting. Nav?
I just had one more quick thing.
Okay.
Could you post the PowerPoint for chapter 4?
No, because we haven't finished chapter 4.
I
Unless
thought we did
not the
today.
chapter that we finished.
Yeah, no, we did chapter 4 today. We're on chapter 5 now.
I have chapter 4 already up there, I think.
Oh, did you put it up there?
It should
Yeah,
be.
it's up there.
Oh,
Thank
okay,
you.
thank you.
I was looking to check myself and not quick enough. Yeah, it's up. All right. All right, Nav, I'm gonna start yours right away. You can stay on if you want. I would
Sure.
rather end it right now.
Yeah.
You want me to end it or you want to stay on?
You can grade it and you will let me know, right? On the comments, like what I missed or,
Yeah, I will put up on the project what you missed.
okay, thank you.
Okay, all right.
Thank you.
All right, see you Thursday.